All right, so we're just going to finish up blood pressure. And then the cardiac cycle, and the cardiac cycle I think is easy. This is what happens after spring break. You all get the dumbs. You get the done, I'm done, and I'm dumb. It's both of them after spring break. And I get the grumpies because all of a sudden everybody's like, I just need you to meet for me for every office hour for the next 3 weeks. I'm like, I literally have never spoken to you before. And in my brain, I'm flipping you off. I'm like, really? Now we're friends? Now? I shouldn't share that with you, should I? Okay. Huh? I can do it in my head. Fine. Okay, so we were talking about blood pressure last time, and we were talking about hypo and hypertension. Okay, all we have to do is go through reasons for hypotension, reasons for hypertension, and what you can do about it. So remember when we talked about hypotension last time, what is the number that is the important one in hypotension? Hypotension. Top, yes. So it's basically the top number, which is the number that is pushing the blood flow out. So we said that mild hypotension, no problem, right? But when your hypotension, when your pressure is too low to deliver adequate blood flow to capillary beds, then you are in what's called shock. And shock means something very specific clinically and physiologically. It doesn't mean, oh, I'm shocked. It means your blood pressure is too low to deliver adequate blood flow to capillary beds. And so, of course, that can kill you dead, right? Especially if you're talking about capillary beds of the brain or the heart or the lungs or whatever. So let's talk about the causes of shock. But you already can figure it out because you know the three things that are pressure related, right? If you're messing with pressure, shock could be caused by something happening with the heart, yes? Something happening with the vessels, or something happening with the volume. Does that make sense? Okay? So let's talk about those causes of shock. After heart attack, right? Your heart's too weak to generate adequate cardiac output, and that can cause shock, right? Does that make sense? Okay? Or what if something causes massive vasodilation, right? Your heart was fine, at least originally, but you get massive amounts of vasodilation. Can you see you would drop the pressure dramatically? And that could happen because maybe I damaged my nervous system so that I couldn't actually give sympathetic stimulation to my blood vessels, and they all vasodilated, right? But the reasons that you'll probably be more familiar with are, what if I have massive amounts of paracrine agents being released that are vasodilators? So, for instance, some little kid gets peanuts who is massively allergic to peanuts, right? And what happens is, for some reason, I don't know what the hell the problem is with peanuts, the peanut antigens cause the release of these massive amounts of paracrine agents because your body thinks it's some kind of pathogen. And so you get paracrine agents, and the paracrine agents just cause generally local vasodilation. But what if you release massive quantities of them into the bloodstream? Then you get vasodilation everywhere, and your blood pressure drops everywhere. Does that make sense? And you go into anaphylactic shock, right? Anaphylactic shock. So anaphylactic shock is just when basically you release so many paracrine agents that they had a systemic sort of effect. And of course, you guys know, what would you do about anaphylactic shock, and why would it work? And tell me why it works. Why would epi work? Vasoconstricts, and two more things. Increases heart rate and stroke volume, right? That makes sense? Do you see that, why that would be an effective thing to combat anaphylactic shock? Okay, and then the other type of shock is, well, one of the other types of shock is toxic shock or septic shock. So let's talk about if someone has a wound and they don't change the dressing regularly, like bed sores, and don't change the dressing regularly, what can happen is these pathogens, or they even could be naturally occurring bacteria. If they are in high quantities, you start to release lots of paracrine agents to allow for vasodilation, to bring in more blood to the area. But now you have so many pathogens and so many paracrine agents that release. As the pathogens go into the bloodstream, they cause paracrine agent release all over the body. you go into septic shock, septicemia, septic shock, and you can die because of that. And of course, most women have heard of it because of toxic shock, because everybody tells you have to avoid toxic shock. Once a woman starts menstruating, she starts using tampons, you have to use them correctly, you have to change them regularly, because the vagina is not sterile. It shouldn't be. It's got naturally occurring flora in it. But the naturally occurring flora gets the balance off because you haven't changed that in a long time. Then you are releasing massive amounts of paracrine agents into the bloodstream, and you can go into toxic shock syndrome. Does that make sense? So one of the reasons that you got to tell your little girls about this, because it says it on the package, but nobody reads the package, you got to tell them that you could actually die associated with that. Or have a limbs removed, amputation.
Is that the same thing that carry your blood pressure?
Massively, yes. Mm-hmm. Go ahead.
Wasn't that big back in the 70s when they were actually sending out tampons in boxes, like to people through the mail, like to try the tampon? So my mom got it.
Oh, really?
And she was really young. And my grandmother was a nurse practitioner, and she's like, no, listen, you have to change this every 4 hours or whatever.
Uh-huh.
And she did, but she got toxic shock. They said there was something in the tampons in the 70s that
I wonder what it
not
was.
in
They
the
probably
tampons
just
now.
reformulated it so that it was less hospitable to bacteria,
Very
I imagine.
And then now, if you use the tampons, don't use the ones that say pearl, because it can cause
Really?
shock.
Interesting. I didn't know that one. Okay. The other thing that can cause massive circulatory system shock is loss of volume. So if you hemorrhage, you can go into shock, right? Because your pressure's too low, right? Or has anybody ever been so damn sick that they lost so much fluid that they could hardly stand up, right? Throwing up, diarrhea, and then all of a sudden you get up because you're like, I gotta go to the bathroom for the 27th time today, and then you pass out because you don't even have enough blood pressure to keep yourself standing. Has anybody else ever done that? Yeah, so damn sick, your blood pressure is so low because of loss of fluid. So those are causes of circulatory system shock. Okay, so now let's talk about hypertension. Hypertension is not one that generally kills you like quickly, like circulatory system shock does, but it kills you slowly and progressively, and it is one of the American diseases. So let's talk about hypertension. With hypertension, again, what number is the problem? Bottom, it's the bottom one, and the top one follows it, right? So when you go into the doctor and you are hypertensive, they'll often say that the cause is idiopathic, which means it's really a lot of different causes, and you only pay $20 for your copay, and I don't want to explain it to you. So let's explain it now. So what are the actual causes of hypertension? Some of them you can't do a damn thing about, genetics, right? I mean, wouldn't you choose different family members, at least some of them, if you could, right? You'd be like, I want rich ones and tall ones who are nice to me. Okay, so you can't do anything about the genetics. You can do something about the other parts. The other biggies are arteriosclerosis and atherosclerosis, which is your blood vessels getting gunked up because of your diet and exercise patterns. That you can do something about. And then vasoconstriction associated with stress, which you can do something about with an asterisk, right? So what happens is arterio and atherosclerosis. This is a postmortem photo, a cross-section of a coronary artery. Let me show you how big the lumen is supposed to be in this coronary artery. This is how big the lumen is supposed to be in that coronary artery. All of the rest of this is arterio and atherosclerosis. This is a calcified plaque. They get crunchy after a while. They're squishy for a while, and then they get crunchy. So what happens here is sort of the American pattern is low levels of cardiovascular activity with high levels of sugar and fat in the diet, right? And what happens there is this starts building up in your vessels when you're in your teen years, and it sort of builds up slowly over time. So let's talk about how hypertension generally occurs. What occurs first is not that your doctor usually tells you you're hypertensive. They usually tell you you have high cholesterol first. But you have zero symptoms when you have high cholesterol. So although the doctor will tell you to change your diet and exercise patterns, generally people don't change patterns when they don't have symptoms, right? Sometimes they'll ask for a pill, but they're not going to change a whole hell of a lot else. Okay, so that's step one. Step two is fast forward a few years and then you go in and they'll say your blood pressure is slightly high. But the slightly high matters because it is the beginning of this arms race that goes on. You start gunking up the walls of your blood vessels, right? Okay? And when you start gunking up the walls of your blood vessels, the afterload in your vessels increases. So what does your heart have to do? Has to pump harder to overcome the afterload, right? That increases the pressure of the whole system. So this pressure goes up, so that pressure goes up, and this pressure goes up, and that pressure goes up, right? So it's asymptomatic for a very, very long time. You're not going to, for instance, start getting kidney damage for a really long time or eye damage for a really long time. But you will be hypertensive. Now the problem is that even though it's asymptomatic, it is doing damage. What kinds of damage is it doing? The kinds of damage that it's doing is it is wearing out your heart, right? It is pushing against massive amounts of afterload, so your heart has to work harder to get the same blood flow. In addition, it's hard on your vessels to be high pressure. If your vessels are under high pressure, they get stretched, they get scarred. When they're stretched and scarred, they're lumpier. They're more likely to get more arteriosclerosis. They're also more likely to catch a platelet. If they catch a platelet, you increase your likelihood of clotting inappropriately. Does that make sense? Okay. So the other thing is that some of your capillaries are really, really susceptible to damage at high pressures. I don't know if you guys know this, but kidney failure is associated with chronic hypertension, as is blindness. So as people age and they have chronic hypertension, the retina, the eye, the capillaries are really susceptible to pressure changes. And so the blindness that is sometimes just associated with being elderly is not so much associated with being elderly. Some of it is associated with blood pressure. Okay? And then you get this risk of stroke and heart attack. What is the risk of stroke and heart attack about? It's two things, really. First off, arteriosclerosis means that your vessels might look like this, right? And then remember, they're lumpier, right? So we've got the double whammy, because I've got a narrow vessel, and then I've got lumpy vessels elsewhere, right? If I have lumpy vessels, they're more likely to catch a platelet. When they catch a platelet, you can form a clot. Clots are not problematic for most of us. We form them, and then they break down because they go through our nice wide blood vessels. But now I've formed a clot, and my coronary arteries are 90% blocked. Or my blood vessels to my brain are 90% blocked. And I get a stroke or heart attack associated with this. In addition to these thrombosis-related strokes, you can also have hemorrhage-related strokes. Any idea why hypertension will increase your likelihood of hemorrhagic stroke? Any idea? Why are you more likely to bust a blood vessel? Because your freaking pressure is too high, right? If you have any weakness in the wall of a blood vessel and chronic hypertension, you're increasing the likelihood. Does that make sense? Okay, so is there any good news here? There's a little bit. So they call this the silent killer, by the way, because you are building up all of the silent symptoms that can lead to strokes and heart attacks. What are the risk factors? And what can you do something about? Stress, because of the increased cortisol in your system with stress and increased circulating nutrients makes these things more likely to have arteriosclerosis, and also stresses vasoconstriction, which increases the pressure of the system. Heredity. If you've got a family history of heart disease, then you need to be nicer to your cardiovascular system than other people. African descent. People of African descent have a predisposition to arteriosclerosis. So again, you need to be nicer to your system. Obesity is a big deal because obesity increases resistance, which means the pressure has to be higher, right? Does that make sense? High sodium, high fat diet. That's a big one. High sodium because of water retention, high fat because of arteriosclerosis. Lack of exercise. If you exercise, you clean out your blood vessels. Okay? And smoking is a huge one. Smoking is fascinatingly related to this story. What smoking does is the current research thinks that the connection is that oxidized cholesterol sticks, okay, and smoking zaps antioxidants from your system. So whatever I eat, if I'm a smoker, sticks to the walls, more likely. So this has proven to be true anecdotally with the evidence that I've done from dissecting a whole bunch of cadavers. from dissecting a whole bunch of cadavers, I can't always tell from the lungs whether someone's a smoker, but I can always tell from the vessels. Because if I cut open an artery from a smoker and squish it, it's like toothpaste. The fat comes out of the wall. So no matter what you eat, it seems to stick to the walls of your vessels when you're smoking. So it's incredibly bad for your cardiovascular system. Go ahead. I don't have enough experience with other kinds of smoking. Typically speaking, so like marijuana, first of all, people don't smoke anywhere near the amount of marijuana that they do when they're smoking cigarettes. Even people who smoke marijuana all the time. Yeah, that's a good question. I don't know whether it does it with the vaping. Okay, so what does your doctor tell you when you have hypertension? Lose weight, stop eating so much sodium, exercise, but don't start an exercise regimen without consulting with your doctor if you're hypertensive. Like, don't go, oh, I think I'll run a marathon to drop my blood pressure because you're going to die. Reduce stress, and then, of course, everybody goes, blah, 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 let's skip to the drugs. That's exactly what happens every damn time. They're like, I could do all of that, but how about you give me a pill? So it should make sense to you that the most common treatments for hypertension are diuretics, yeah, to drop the fluid volume. Vasodilators, which include beta blockers and also calcium blockers. Beta blockers decreases cardiac output, it drops your heart rate, but it also allows for vasodilation, and calcium blockers do the same thing. And then ACE inhibitors. What do ACE inhibitors do? You guys remember ACE? Angiotensin converting enzyme. So if you don't have ACE, what do you not get? Angiotensin 2, which is the most powerful vasoconstrictor in the body, and you also don't force aldosterone release. So ACE inhibitors are common treatments. That make sense? Okay. I bet those of you who work in clinical situations have used all of those. Yeah? Go ahead.
But I think, isn't that true, like once you get to that point, you've already done like a ton of damage.
Oh, once you're getting to the point that you're actually being medicated in this way?
Once you get to that point where the doctor's telling you, okay.
Yeah, you've already done tons of damage. It is not irreparable, but it is not reparable with medication. So you can do a tremendous amount for repairing your cardiovascular health if you change your diet and exercise patterns. This is just managing symptoms. This is not actually managing the cause, right? You're going to have to change your diet and exercise patterns. You're going to have to change the way that you eat. You're going to have to, like, with the doctor's supervision, get more active so that you can clean out your blood vessels. Just that there is no homeostatic mechanism. There's no medication that is as effective as your homeostatic mechanisms. There just isn't. Okay? All right, so that's pressure. Now what we are going to do is we're going to move to heartbeat coordination and the cardiac cycle. And if the anatomy is familiar to you, then this phys will be pretty easy. So let's just go back and review stuff that you have known before, and then we'll get into the cardiac cycle. Okay, so stuff that you've done before, and we actually have already talked about it again. But as far as the anatomy in relationship to the electrical activity of the heart, you guys know that the SA node gets to be the boss, right? And the SA node will depolarize, right? And send the electrical stimulus, the sodium and calcium, through the atria from the top to the bottom, right? And then the atria will contract. Everybody good? Top to bottom. And then you also know that right about the time the stimulus gets to the bottom of the atrium, it will take over what the AV node was going to do. The AV node was going to generate an action potential anyway. It's just that the SA node gets to boss it around. But please remember, this is important. The AV node does not talk to the contractile cells. Remember, the AV node only talks to its buddies, the AV bundle. Those are pacemaker cells. The AV bundle talks to the left and right bundle branches, still not talking to contractile cells. And it's only the Purkinje fibers that talk to the contractile cells, right? Do you guys remember that? And the Purkinje fibers talk to the contractile cells from the bottom and the sides of the heart first. So what happens is the atria contract top down, but the ventricles contract bottom up. And also, this forces a lag time between atrial contraction and ventricular contraction. At a resting heart rate of around 75 beats per minute, the lag time is about 0.2 of a second. So you go atrial contraction, pause, ventricular contraction. Does that make sense? Okay, and that's important for ventricular filling, yeah? It's also important for delivering blood to the wall of the ventricle. Okay, so keep that in mind. Everybody good? You all right with that? Okay. Nikki says that every time I ask that, you aren't. And, you know, I literally cannot read your mind and wouldn't want to if I could. So, because you've all been shooting daggers ever since we started the endocrine system, honestly. It's just like, I prefer not to know what you think of me. It's really none of my business, is it?
I like it when you say something and you go, did you get it? You get it? And I'm like, Melanie, I did not get it.
Well, I am asking for a reason, jerk. Okay, so what we need to do now is we need to put the electrical activity together with the mechanical activity of the heart. What do I mean about electrical activity, mechanical activity? Electrical activity is depolarization, repolarization, right? Mechanical activity is when the chambers contract versus relax and when the valves open and close. So all we're trying to do is make it into one story, right? We're trying to make it into one story. You depolarize the atria and then you contract the atria. And then when are the valves open and closed and all that kind of stuff. Does that make sense? So they call this process, this whole story, they call it the cardiac cycle. And it's actually pretty logical. Okay, so before we look at the bottom part of it, let's just look at the top. This is one of those figures that it's really hard to get when you project it, so you will want to look at this one at home, but it's not too bad. So it is a cycle. I'm going to go through it simply first, and I'll add some detail, and then I've got an animation to show you guys, okay? All right, so what we're going to do is we're going to keep track of when chambers contract and relax and when valves open and close, okay? Then we'll go back in and add heart sounds when you hear the lubbed up, right? And then we'll coordinate it with an EKG. Good? Okay, and it's going to make sense. Okay, it's a circle, so it's completely arbitrary where you start, completely arbitrary. Your textbook happens to start right here when the AV valves are open. And see if this makes sense to you. They call this part when the AV valves are, this phase when the AV valves are open, they call it ventricular filling. Ventricular fillings. Does that make sense? The AV valves are open, so the ventricles are filling. Correct? And please look, this is the length of the phase. It's a decently long phase, this ventricular filling. Okay, so I want you to think for a second. The AV valves are open, the ventricles are filling, right? That ventricular filling is going to end when what valves do what? AV valves close, right? And let's talk about why the AV valves are going to close. The AV valves are going to close when the ventricles start to contract, right? And increase the pressure on the backs of the AV valves. Does that make sense? Okay. So notice this period right here. This is ventricular filling. It ends when the AV valves close. That starts this brief little period right there. Do you see number 2? That brief little period is called isovolumetric because no fluid is moving anywhere. Isovolumetric ventricular contraction. Why is no fluid moving anywhere? Because the AV valves are closed and the semilunar valves haven't opened yet. Does that make sense? Okay? So, little brief time called the isovolumetric ventricular contraction. Isovolumetric ventricular contraction is going to end when what valves do what? AV already closed. Semilunar valves are going to open. Does that make sense? Okay. When the semilunar valves open, we are in this next phase, which is pretty long. Do you see it right here? This is called ventricular ejection. Does that name make sense to you? Ventricular ejection, so blood is leaving the ventricles. Okay? And it's a decently long phase. You're pumping blood out at that point into the aorta and the pulmonary trunk. Good? Okay? Ventricular ejection is going to end when what valves do what? Semilunar valves are going to close. The reason the semilunar valves close, by the way, is the ventricles are empty and relaxing, low pressure, and the arteries are full, high pressure, and tries to go backwards for a second, right? And gets caught in the cusps of the semilunar valves, right? Okay, so that is called ventricular ejection. It will end when the semilunar valves close. And notice there's this little bitty part right here, pretty short again, during which the semilunar valves are closed. The AV valves are still closed from the last time. This period is called isovolumetric ventricular relaxation. Isovolumetric again, because no blood is moving anywhere. This time the ventricles are relaxing. that make sense? Okay, so isovolumetric ventricular relaxation is going to end when what valves do what? AV valves open. Yes? AV valves are going to open, and you're going to start the whole story again, right? And when the AV valves open, you start ventricular filling, right? The only thing that I didn't add the last time is what is the relationship of atrial contraction and ventricular filling. A lot of people have a misconception. Does anybody know why the AV valves actually open? Anybody know why they open? It's the pressure because of what? Yeah, it's the pressure because of venous return, right? So you've got this continuous venous return, and as soon as there's enough blood on top of the AV valves, that opens them. But a lot of people are in the habit of thinking that the atrial contraction is what causes the AV valves to open. It is not. The AV valves will open in the absence of atrial contraction. So what does atrial contraction have to do? So as I get venous return back in, right, and the venous return builds up pressure in the atria, that causes the opening of the atrioventricular valves. And then 80% of the blood goes from atria to ventricles, right? And what does the atrial contraction do? Squishes the last 20% in. Does that make sense? Okay? If it was atrial contraction that was responsible for AV valves opening, I would be dead because I have atrial fibrillation. With atrial fibrillation, instead of doing this, this is what a heart should do. Atrial contraction, pause, ventricular contraction. Atrial contraction, ventricular contraction. Atrial contraction, ventricular contraction. If the atria were responsible for opening the valves, and my heart does this, so this is, I can't do it, I'm not coordinated. So imagine that the ventricles are going to continue doing this. This is what my atria do. Can you see that I would die if I wasn't getting any blood into the ventricles? Does that make sense? So what I want to look at, and then we'll look at an animation, is tell you a rule, and that is that you can never have both sets of valves open at the same time. So if the AV valves are open, you know what the semilunar valves are doing, right? Do you see why you can't have that? Because you'll get backwards flow of blood. You can have them all closed at the same time. That's okay. But you can never have both sets open at the same time. So if you know the position of the semilunar valves is open, then you know the position of the AV valves is closed, right? And you don't have to memorize it, right? Okay. And then the other thing is, what about heart sounds? The lubbed up, lubbed up, lubbed up. What does that have to do with this story? It is the closing of the valves and the blood slamming against them, right? So there's two sounds, lubbed up, lubbed up. The first one is lower. The second one is higher and slightly more staccato, like sharper, okay? So the lub is what closing? AV valves closing, right? And then the dup is semilunar valves closing. Does that make sense? Are we good with that? Okay. So last thing, and then we'll look at the animation, which will reinforce this. So here's the heart sounds. Notice where they occur, right? Isovolumetric ventricular contraction is the beginning of the first heart sound. Isovolumetric ventricular relaxation is the beginning of that one. And the only other thing is, can you put it together with the EKGs that you did in lab? The EKGs that you did in lab told you that the P wave, right, corresponded to atrial depolarization, which would precede what mechanical activity? It precedes atrial contraction, right? Which is at the end of ventricular filling, okay? And then the QRS complex is what? Ventricular depolarization, which precedes ventricular contraction. Does that make sense? Okay, so let's look at the animation. I uploaded this for you guys already, but
ventricular contraction causes the atrioventricular valves to close,
okay,
which signals the beginning.
Before he goes any further, okay, so what he just did was, do you see those atria just finished contracting, right? Do you see that? And now what's going to happen is the ventricles are going to start contracting.
Ventricular contraction causes the atrioventricular valves to close, which signals the beginning of ventricular systole.
Okay, so Right here, we are in isovolumetric ventricular contraction. Why isovolumetric? Look at the position of all 4 valves. They're closed, right? He calls it volumic and he says systole instead of contraction, but it doesn't make any difference because you guys can do that, right? Okay, so predict how this is going to end. This phase is going to end with what? Semilunar valves opening, exactly.
The semilunar valves were closed during the previous diastole and remain closed during this period. Continued ventricular contraction increases pressure in the ventricles above the pressure in the aorta and pulmonary trunk, causing the semilunar valves to open.
That is ventricular ejection, right? We did isovolumetric ventricular contraction, then ventricular ejection. How's this going to end? Semilunar valves are going to close.
When the ventricles relax and their pressures drop, blood flowing back toward the relaxed ventricles causes the semilunar valves to close, which is the beginning of ventricular diastole.
That is isovolumetric ventricular relaxation, which is going to end when AV valves open. Good.
Note that the atrioventricular valves remain closed. When the pressure in the ventricles becomes lower than the pressure in the atrioventricular valves open and blood flows into the relaxed ventricles.
Ventricular filling. What's going to happen at the end of ventricular filling? Atrial contraction. Good.
This accounts for most of the ventricular filling. The atria then contract and complete the ventricular filling.
Isovolumetric ventricular contraction, ventricular ejection. Isovolumetric ventricular relaxation, ventricular filling, atrial contraction. You guys okay with that? Okay, so if you look at your notes, it just goes through a few things. I'll do arrhythmias and fibrillation in just a second. But please note that when you talk about ventricular filling, there's not a whole lot of memorization. And with ventricular filling, what is intuitively the position of the AV valves? Open. That means that the semilunar valves are automatically closed, right? Okay, that makes sense? At the end of ventricular filling is when atrial contraction is going to occur. Then we go into the next stage, which is isovolumetric ventricular contraction. If it's isovolumetric, what's the position of all of the valves? Closed. You got it? Okay. Isovolumetric ventricular contraction is going to end when the semilunar valves open, right? And when the semilunar valves are open, that's ventricular ejection. What's the position of the AV valves? Closed, exactly, right? Okay. And then you are going to go back into isovolumetric ventricular relaxation, and the position of all of the valves is what? Closed, exactly. Good. Okay, so it's not that hard, right? Do you see? So all you have to do is make sure that you know the terminology because you already know the story, right? Okay, so last question, and then we'll talk about some abnormalities. So the last questions are, can you put together the mechanical and the electrical activity and then figure it out? Okay, so the mechanical activity of the heart must be coordinated with the electrical activity of the heart. So for each event, can you list the corresponding mechanical activity that would follow? So the first event, the electrical activity, is the atrial depolarization as represented by the P wave. The mechanical event that immediately follows that is what? Atrial contraction, yes. And so the question after that is, does that mechanical event cause the opening or closing of any valves? Does not. Doesn't. Okay, next one. The electrical event is the ventricular depolarization as represented by the QRS complex. The mechanical event that immediately follows that is what? Ventricular contraction. And does the ventricular contraction cause the opening or closing of any valves? Yes. There are 2 of them. Get them in the right order. Closes AV, opens semilunar. Got it? Good? Okay. So let's talk briefly about the fact that if you have abnormalities of the electrical activity, you can get abnormalities of the mechanical activity, right? So what it should do is atrial contraction, ventricular contraction. Anything other than that is an arrhythmia. Arrhythmia is a general term, right? it means you're doing something other than this pause, this pause, this pause, this pause, right? So it's just a general term. One of the forms of arrhythmia is a fibrillation. And with the fibrillation, I showed you what an atrial fibrillation is, right? Ventricles are doing their job just fine and the atria are spazzing out, right? What about ventricular fibrillation? Ventricular fibrillation is a much bigger deal because the atria are doing just fine, but they're not the pumping chamber. So they're doing this. What are your ventricles doing? They're spazzing out, right? And ventricular fibrillation can lead to ventricular tachycardia, which can lead to death. Okay, so that's a big deal. So there are a whole bunch of other forms of arrhythmias. One of them is when an ectopic pacemaker, something tries to launch a coup and take over the heart rate from the SA node. And it's just like, oh, I'm over here in the right ventricle, and I'm going to try to start the electrical stimulus from the right ventricle. What happens is then your chambers are contracting when they're not full, and the right side's contracting independent from the left side. What do they do in situations like that? They can either give you an artificial pacemaker, or they can do an ablation where they burn off those cells that are misbehaving. They can do either one of those things, right? Okay, so when you guys go into clinical training, you'll learn a lot more abnormalities and arrhythmias, but I've got a fun thing to show you. This you're not going to be tested on, just for fun. Have you guys seen this one? Oh, I got to turn this back on. I can't have the Wi-Fi and the clicker program running at the same time, so I have to like turn it off, and then the clicker program does not like Wi-Fi. Give me a second. Just wait right there. Come on, you bastard. Okay. Guys ready? So these are the atria and the legs are the ventricles. So sinus rhythm is normal. He's the cutest old man, I love that. 6 sinus.
Wow! Come sera Ethelyn, la mer la plage?
This is me. He's so cute. This one's good. Come on. There is literally no disco fibrillation. They're so cute. Isn't that adorable?
Oh man, do some of those moves, man.
Huh?
Yeah.
I was showing that to my husband last night, and he's like, what? Why would you? He doesn't think anything about physiology is interesting, and he's so wrong. Hate how wrong he is. Okay, so we're going to do a quiz. It's just short. Yes. It's not hard. There's only 6 questions. Okay, pay attention. This is an important term right here, right there. Ready? Start.
Any hints?
A or B?
Very helpful.
My channel's not,
41.
I know what it is, but it's not working.
You can write it down if you want. You just,
On it, but it's not working. Why did it say that?
no, don't. You know, I'll get mad at you.
We share answers.
You sure you trust English people?
Nope. Nope.
I wouldn't.
My Mississippi person, no, my Southern person.
Mine's not working either. I don't know why.
I don't know how to work those. If anybody knows how, they can help her. Okay. Click in, Sam, click in.
I did.
Okay. No, you didn't.
I think I did. It says I did.
Okay, good. All right. So, hey, you guys did better than the other class. Local, if you detect those things in a capillary bed and we release paracrine agents, we're not talking about systemic regulation, we're talking about local regulation, then it should cause dilation of the local arterioles leading to that capillary bed. Now, contrast that with this one. Write it. Don't take pictures.
Right.
Write it down. Huh?
Oh.
Yeah.
I know this one.
This one? What are you talking about?
I think. Right?
What?
Right?
I don't know. Start. Who's bringing snacks to the study session?
Whoa.
You were supposed to.
Oh, yeah. I need to make it one more.
You better make that run then.
I've got a bad heart, man.
Dude, you said you were going to.
Because Melanie said I wasn't getting extra credit points, so.
No.
Okay, so low pH if detected in the aortic arch would cause the medulla oblongata to activate the sympathetic nervous system. Okay, this is systemic regulation, not local regulation. Okay, number 3.
I got this.
You do? Start.
2 for 3 isn't bad.
Ooh, look at you guys. Yes, it is. That's the answer. Good. All right, let's see how you did with cardiac cycle. It's not bad. It's not de-erring. It's during. Ready? Start. Very good. Yes. Hey, you guys got cardiac cycle, right? It's not that hard. See? What? It's hard? Vivian. It's because you guys are all, your brains are done this semester. You're like, I'm done. We gotta get through the end together, guys. Ready? Start. There's only one more question. There's only 6 questions. I'm getting a thumbs down from Daniel today. I don't know if you guys noticed.
And no treats.
You just remember that before I write your test. I haven't written your test yet. I'm going to write a special one for him.
Bring treats. And whatever she
Writing
likes.
the dumb test for me? Is that it?
Okay. Yes, it is these. A and P does the opposite. Last one. Start. I need at least 50% correct on this. Only 2 answers. Ooh. Ouch. Okay, so let's talk about that. Renin is released in response to low blood pressure detected at the kidney. Okay, renin interacts with an inactive plasma protein. What is it? Angio.
You say it wrong last time.
That one.
Something happened over here.
I know what it is. Hey guys.
Angiotensinogen from where? Liver. Okay. Renin interacts with angiotensinogen from the liver to make what? Angiotensin 1. Angiotensin 1 interacts with what? ACE from the lungs. Yes. And the ACE and angiotensin 1 make angiotensin 2, which does what to your vessels? Vasoconstricts to bring up the pressure and causes release of aldosterone. Right? So you knew that, right? You actually knew this one. Okay? I mean, it was in there somewhere. Okay. You're done with that one, but we're going to start on blood.
Real quick, so for the...
Hold on, let me save.
For that one question about local,
Is that like auto-regulation?
Yes. Mm-hmm.
Question 2 was
That you guys got lucky because that quiz got captured on the recording, so it's on there.
Well, that means I can change 2 of my answers.
Yeah, I'll sleep on.
very nice.
Mm-hmm. See, Daniel, somebody likes me.
Whoa, wait. I like you.
Okay, this stuff is not on your 3rd test. It's on your 4th test, and blood's pretty easy. I know you think I just say that. Here, you usually, if you were on some kind of clinical track, will cover blood in like 2 or 3 different contexts in different classes. When you take micro, you're going to do immune system functionality. If you take cell, you'll also do more immune. Didn't you? Right? Yeah, because he does a bunch of that stuff. In anatomy, I usually do blood, although if I run out of time, I get rid of it. But what Phys does that the other classes don't usually do is blood clotting. Okay? So the only really new part in this, if you've had blood other places, is going to be clotting. And it's not very hard. It's not super easy, but it's really the only difficult part. So here's the deal. I'm going to start on it today. I'm not going to do anything super difficult today. I am on Wednesday going to upload an assignment for you guys to do between Wednesday and Monday. For you to practice with clotting before you come in. It's not hard, and we will probably finish the blood clotting notes about 20 minutes in, and then we'll start on, is it urinary first or respiratory?
Respiratory.
I hate respiratory system. We'll start on stupid respiratory system. We'll start on respiratory system. So we're doing fine as far as timing goes, and blood, we're not doing hard blood stuff today. Don't worry. Doesn't mean you can, I mean you can leave if you want to, but I'm still going to talk. Okay, so blood. You can talk, if you want to, about all the types of white blood cells and the red blood cells and the platelets and the blood plasma. You're going to do that in other classes, so what I'm going to do is I'm going to tell you a little bit about what blood plasma is like. Then we'll skip into how red blood cells are formed, right? And the whole EPO story with red blood cells. And then after that, we'll go straight into blood clotting. And there's not really that much content in here. Okay, so first thing is just on the before you begin, the only thing that I want to talk about is blood plasma. Blood plasma is 90% water, but it has some important functions. Basically, anything water soluble that you need to carry from point A to point B, you carry in blood plasma. Like all your ions and your water soluble hormones are going to travel in the blood plasma. Nutrients travel from absorption to all cells, and waste products travel in there. So that's the blood plasma is carrying most things around. There's only a few things that actually travel inside the cells. Like oxygen travels inside red blood cells, right? Does that make sense? But most stuff actually travels in the blood plasma. So just a teeny bit about blood plasma. In addition to nutrients and ions and waste products and that kind of stuff, there's also some proteins that are kind of unique to blood plasma. One of them is albumin. Albumin is made by the liver. Albumin is important because remember how I told you that lipid-soluble stuff needs an escort protein? Yeah? This is a very common escort protein for lipid-soluble stuff. In addition, because albumin is in high concentration in plasma, it helps to maintain your blood volume because remember, you push stuff out, you push water out of capillaries because of filtration, right? And then you pull it back in because of a high concentration of solutes, and this is one of them. Albumin is really helping you maintain your blood volume. Okay, and then the other ones, clotting proteins, we're going to talk about in a lot of details, and then the globulins. They literally named these things globulins because it ends in IN, which means it's a protein, and it was kind of glob-shaped, and somebody goes, I'll call it a globulin, right? And you've got alpha and beta globulins. Those are also escort proteins for lipids. And then gamma globulins, have you guys heard of gamma globulins? Usually in Utah, I've had better luck with people knowing about gamma globulins. Gamma globulins are antibodies. You make them yourself when you are exposed to pathogens. They're part of your immune system. But if, for instance, I was going to go from Utah to like a really rural part of Central Asia, I wouldn't have been exposed to the pathogens there, so I wouldn't have antibodies for them, right? I've never been to those areas before. So what I could do to give my immune system a little artificial buffer is I can give myself a gamma globulin shot. And the gamma globulin shot is the antibodies for the pathogens I think I will meet when I get there, right? And the reason that people usually know more about these in Utah than they did in Texas is because we have a pretty big military community and also missionaries, right?
I got one before I went to Argentina.
You did? You got a gamma globulin shot? I heard they hurt like hell.
Hurt like hell. Sure
Yeah, they call them peanut butter
Peanut
shots.
butter.
They're super viscous.
Charlie Wars, you know.
Yeah, and they're different for different parts of the world. So if I went to sub-Saharan Africa, I would get a different one than if I went to Central Asia or Argentina. I don't need one to go to Canada, right? Or Mexico, because we've all got the same germs pretty much. So anyway, that's what gamma globulins are. And really what they're doing is they're just giving you an artificial window of protection while you make your own antibodies. That's what they're doing. Okay, Then the clotting proteins. Clotting proteins we'll talk about in detail next time. And then all of this other stuff is traveling. Go ahead.
Do you need to do that when you like go to like, or is it just certain like rural areas like you said in Gator? Do you need to go to any?
It's not when you go any place. It's when you go someplace that's like got distinctly different pathogens than the one that you're actually used to. Although, interestingly, because we're such a mobile population right now, a lot of the antibodies are getting really shared globally. But if you go to really, really rural places, a lot of times you're going to need a gamma globulin shot. I've never had one, and I've been to Africa, but only to the northern parts of Africa. I've been to a bunch of the Middle East. I've been all over Europe and South America, and I've never had one. No. Maybe, I think it's just because the first thing I do whenever I get anywhere is eat street food. I'm just like, I'm going to build up my immune system. Street food's the best food, man. It's like amazing, and you can buy like tanks of it for like $3. It's the best thing ever. Okay, so general information. So let's just talk about blood. Blood is thicker than water, although I've always just kind of thought that phrase was silly. It's about 5 times as viscous as water. It's like whole milk. It's also warmer than the rest of your body. It's 100.4. It's usually deep, so it stays a little warmer than the rest of your body. It's kind of sticky because it's got all kinds of sugars and proteins in it. And its pH is really, really tightly constrained. Look, its pH is 7.35 to 7.45, and if it goes much above or much below that, you are going to start changing the shapes of your proteins in your blood. So one of the last 2 body systems we're going to do are respiratory and urinary. And respiratory and urinary are your primary 2 body systems that regulate your blood pH. Respiratory because it has the capacity to decide how much carbon dioxide to blow off, right? And carbon dioxide is directly related to pH, right? And urinary because it can decide how many H pluses to pee out, which is directly related. So those 2 are primarily going to regulate your blood pH. And then it is isoosmotic or isotonic to about 0.9% saline. So if you use normal saline, that's what they're talking about. It makes up about 8% of your total body weight. Males usually have around 5 to 6 liters and females have around 4 to 5, but it's just based on body size, average body size. So like a big woman is going to have more than a very, very small man. Okay, so what does your blood do? Well, you know what it does. It's basically carrying things from point A to point B. Nutrients from absorption sites to all cells, waste products to kidneys, hormones from whatever released it to the target cell, all of that kind of stuff. In addition, it's also helping you regulate your pH because it's got buffers in it. It's also helping you regulate your fluid volume because it's the only fluid that can clot, right? None of your fluids can clot except that one. It's also protecting you from pathogens because it's got antibodies and white blood cells and all that kind of stuff in it. And it is stabilizing your body temperature because it takes blood from really, really warm organs. For instance, if your liver was really, really metabolizing, it'd be really warm, and takes it to other places and cools it down. So it's like an air conditioner, basically. And then it also has the capacity to like homeostatically level out your ions. So for instance, if your brain needed a whole bunch of calcium, it just takes it from your bones, right? Takes it from one place to the next. Okay. So let's talk about red blood cells just a little bit. So hopefully you learned in anatomy the process of red blood cell production is called erythropoiesis. Erythropoiesis. And erythropoiesis occurs where? Where do you make red blood cells? Yeah, you make it in the red bone marrow. And so let's talk about the process. So you do a lot more red blood cell synthesis than you think. Your red blood cells only live about 120 days. They get the crap beat out of them as they're going through capillaries. And so you are consistently sort of making new red blood cells and you're trashing the old ones. So one of the things that happens, do you guys remember that the liver has these wide sinusoid capillaries, these wide capillaries with big holes in them? So as red blood cells go through the sinusoid capillaries of the liver has these macrophages that are like sitting on top of the sinusoid capillaries, and what they're doing is they're looking down to see if that red blood cell looks like it has one more round left in it, and if it doesn't, it recycles it. So if it comes through and it looks like it's been rode hard and put up wet, which is a Texasism. Do you guys know that? Anybody ride horses? I rode horses when I was a kid. So yeah, rode hard and put up wet. Then it recycles it and recycles all the good parts. So we'll talk about the recycle mechanism in just a second. So now, in order to do erythropoiesis in the red bone marrow, one of the things that you have to have is iron. And you guys, of course, know that iron is one of the components of hemoglobin, right? Hemoglobin's primary function is to carry oxygen gas around, and it carries it on the heme portion of hemoglobin. Like a little yellow portion with the iron in the middle, that's the heme portion of hemoglobin. So, in order to make red blood cells, you need iron. You need iron in your diet. We'll talk about where you get it, and we'll talk about how you lose it, right? So, you are going to need iron, and the major sources of dietary iron are going to be meat, liver, talk about that in just a second, shellfish, egg yolks, beans, nuts, cereal, leafy green vegetables, but you're always losing it, and you lose it anytime you're losing fluids. You have the capacity to be losing it, so you lose it via urine, the skin, and women lose a lot of it via menstruation. So you are far more likely to be iron deficient if you're a woman in your reproductive years than if you're a man or if you're a woman out of your reproductive years. So the thing that happens here is that, for instance, if you are iron deficient, you can increase absorption. So a woman immediately after she menstruates, she would absorb more iron in her intestine, right? Her absorption rate would go up. That makes sense? Okay, so you can change the absorption rate based on the body's needs. If you ever have extra iron, you store it in your liver, which is why liver is a really, really rich source of iron. So when I was a kid, I was really anemic. I don't know why. I was like 5 and super, super anemic, and my mom would constantly try to get like leafy greens and egg yolks and all that crap, and I ate it. But in addition, I loved liver when I was a kid. She would like fry up chicken livers and stick them on toothpicks on the bottom shelf, and I just like toddle on in there and eat some chicken liver, which sounds absolutely repulsive to me too now, considering I know what the liver does. But I'm not anemic anymore. Thanks, Mom. So yeah, the liver stores lots and lots of iron. Okay, let's talk about a couple other things you need. They are not part of the picture, but in order to do DNA synthesis, which you are going to need to do cell division to make red blood cells, right? That makes sense? DNA synthesis before cell division, you need folic acid or folate. And folic acid or folate is actually necessary to make the thymine in DNA. And you get folic acid from green plants, yeast, liver, and citrus. And folic acid is a big one because it's not just needed for DNA synthesis for red blood cell production, it's also, it's also super important during pregnancy. About 20 years ago, they figured out that one of the major causes of neural tube defects in women, pregnant women, neural tube defects meaning your nervous system starts out flat and it's supposed to close. And so you have a central canal and the ventricles of the brain. And if you don't have enough folic acid, it doesn't close completely, and so you can get spina bifida and some other neural tube defects and hydrocephalus. So about 25 years ago, they started a major information campaign to let women know that they needed to increase their folic acid intake during pregnancy. But the thing is, and this is worth knowing because a lot of you guys are young enough that you are still going to potentially have children, that you actually need folic acid to be high during your first 2 weeks of pregnancy. How many of us knew we were pregnant during the first 2 weeks? No, I did not know I was pregnant during the first 2 weeks. So the important thing to know is if you are sexually active or could become sexually active and would not terminate a pregnancy, you need to be watching your folic acid, right? You get one chance to put together a nervous system, and that's it. So luckily, it's pretty easy to get. Drink some orange juice, eat a salad, you'll be cool. Also, it's in every single multivitamin for women has high levels of folic acid too. B12 is also needed. B12 is needed for DNA synthesis in all cells. Now, that's not generally a problem for many of us, except if you're a vegan. If you are truly vegan, B12 levels are really low in plant sources. You're going to have to be very careful about what you're eating, especially during pregnancy. It's not like you can't have a healthy pregnancy. You can, but you're going to have to pay attention. Okay, and then the one we want to get to is EPO, or erythropoietin, which we already talked about a little bit in the last set of notes. EPO, or erythropoietin, is produced from the kidney in response to hypoxia, or low levels of oxygen. And let's look at what it does. If the kidneys detect low oxygen, here's what they do. They secrete EPO. And erythropoietin, EPO, is going to target the place that can do erythropoiesis, which is where? Red bone marrow. So EPO is going to go to red bone marrow. And it is going to cause erythropoiesis. Erythropoiesis. Yeah? And then erythropoiesis is going to do what ultimately to your level of oxygen? Increase your level of oxygen, right? That's a nice little feedback loop, right? So before we go any further, what category of hormone regulation does this fit in? Hormonal regulation, nutrient or ion source, or nervous system? Yeah, this is like non-hormone nutrient or ion source, right? It's not a hormone that's feeding back, right? Does that make sense? So this is one that fits in there. So what it does is it causes your red blood cells to do erythropoiesis. When you are doing erythropoiesis, you can make 2 to 3 million red blood cells per second. You can do some serious work when you are doing it. So let's talk about what are some possible causes for this to occur. Why would you be low oxygen? That hypoxia could have been triggered by hemorrhage or destruction. Or what if I just gave blood? If I give blood, doesn't it make sense that your oxygen content is going to go down? And that will trigger the release of EPO and cause erythropoiesis, right? And then give yourself about 2 weeks and you'll regenerate your entire blood volume. So you're cool, right? Okay. Also, destruction. So malaria, I don't know if you guys know, malaria is an intracellular red blood cell parasite. And what it does is it goes inside your red blood cells, and during part of its life cycle, it causes destruction, right, of the red blood cell as part of its life cycle. So it drops oxygen content. That's part of the reason that people with malaria are exhausted. Does that make sense? Okay, so you will make way, way high levels of EPO if you're infected with malaria. Or decreased availability of oxygen. So COPD is chronic obstructive pulmonary disease. It's anything that causes problems with getting air in. That will cause this. Or go to high altitude. Be a little girl from Texas who moves to Utah, right? And I had plenty of red blood cells for low altitude because there's lots more oxygen availability. But when I moved to high altitude, I didn't have adequate red blood cells. I couldn't even walk up the freaking stairs without getting winded in Utah when I first moved here. So what happens is I'm going to release some EPO and give me a couple weeks, and then I can increase my red blood cell synthesis enough, right? That makes sense? So that's the high altitude. Or what if you're just really active? If you are a really active person, your skeletal muscles are gobbling up oxygen, you are going to have significant and consistent signals that you need more oxygen. So people who do a lot of cardiovascular activity actually have more red blood cells than the rest of us because they need them, right? They have greater oxygen carrying capacity. Okay, so let's talk about blood doping. Okay, so I know you guys have heard of blood doping, and it has to do with this story. So one of the things that we figured out, not me, but them, is that EPO could be really helpful for getting over some particular clinical situations. Like if I had chemotherapy because of leukemia, it doesn't just trash the good cells, it actually trashes all of the cells. And part of the problem with healing and overcoming the cancer is that you have reduced oxygen carrying capacity. Well, what if I shot you up with some EPO? It's protein, so you have to actually inject it. So what if I shot you up with some EPO? Then you could increase oxygen carrying capacity and maybe heal better, right? And so this is a drug sometimes sold as Procrit. Have you guys ever heard of Procrit? Okay, so it helps with getting over certain clinical situations. It's very useful. We didn't used to be able to make it in the lab. Fascinatingly, before about 10 years ago, the place where we got EPO has questionable human rights history to it. We used to buy the urine of malaria patients from countries that had high levels of malaria because they have so much EPO that it ends up in high quantities in their urine. So we would buy it from countries with high levels of malaria infection, and then we would cleanse it and we would use it. I really hope that we gave them some medication, but I can't really depend on us. I mean, I've met us, right? No. So anyway, that's where we used to get it. Now we actually can make it in the lab, and now that it's more readily available, it's being abused, right? Because with elite athletes, they train at high elevation so that they can get that advantage. But when all of them do that, it presents no advantage, right? They all do the same thing. You're all training in Park City, right? So then what do you do next? Well, you say, okay, well, I'm going to, before I start training, I'm going to give my own blood sample and keep it in the fridge from clotting, right? Keep it from clotting. And then 2 weeks later, after I've regenerated my own blood volume, I'll stuff my own red blood cells back in, okay? That's one form of blood doping. And the other form of blood doping is just to shoot yourself up with EPO. And the problem is your blood can become too viscous to pump effectively, right? And so you can have heart attacks and strokes. It's called heart attacks and strokes. It's called polycythemia. So now what they do in elite level professional sports, like cyclists are famous for blood doping. They just love some blood doping. Lance Armstrong said that he did it. Now what they do is they say, we will give you this range of red blood cells that you can have in the sample, and I don't care how you get higher than that, you're not competing. So, and it's a really generous range. It's like not the range that any of us would get into because we're not elite cardiovascular athletes. So they say, I don't care how you got higher than this, then you're not competing. And males have a higher amount than women because we'll see testosterone actually has an impact as well. So that's blood doping. So testosterone is an interesting one because testosterone is another trigger for EPO release. So testosterone causes you to release EPO. So per cubic millimeter, which is about the size of a big drop, the range of red blood cells is about 4 to 6 million in people. But look, males, their average is about 5.4 million per cubic millimeter, and females have about 4.5 million. So males have greater oxygen carrying capacity than females. Does that make sense? Okay, so once you get up to the level of elite athletes, so we're talking about Serena Williams versus, give me a male tennis player.
No. You want board?
He's dead. Is he dead? Rafael Nadal, no. Okay, this dude. What? Roderick, oh yeah, okay, Federer. So those two, both badasses, right? Incredibly good at their jobs. Do they compete against one another? Generally, no. Testosterone has greater oxygen carrying capacity, and also it causes more muscle mass building. Does that make sense? So you can whoop his ass at chess, but the elite, and she would kick all of our butts, right? I mean, she would kill us all. But because of the increased oxygen carrying capacity because of testosterone and the increased muscle mass, it really does matter once you get up into the level of elite sports. Okay, so during development, before, when red blood cells are actually being made, they actually will eject their nucleus. So red blood cells, you know what they look like. They're, you know, these biconcave discs. And they don't have a nucleus. They've lost most of their organelles, but before they do that, they actually make boatloads of hemoglobin. So what they really are is little donuts filled with hemoglobin. And so they make hemoglobin. Hemoglobin is that red iron-containing pigment, and it makes blood red. Blood is redder when oxygen is bound to hemoglobin than when it is not. And males and females have different amounts of hemoglobin as well, because if you looked at 100 mils of blood and dried out the amount of hemoglobin, because testosterone causes red blood cell synthesis, you can see that males have significantly more hemoglobin in the same quantity of blood. That means greater oxygen carrying capacity as well. Okay, so we will stop there for today.
So men's blood is redder than women's?
Men's blood, when oxygenated, could be redder than women's. Yeah.